Let's now look at the organization of a sarcomere. A sarcomere is the functional contractile unit of a skeletal muscle fiber. And remember, a muscle fiber refers to a single muscle cell. A sarcomere is defined as the distance from one Z-disc to the next adjacent Z-disc. And myofibrils contain multiple Z-discs. Thus, there are numerous sarcomeres in each myofibril. Now, I want you to think about our second floor of our building. And if you think of the entire floor as a myofibril, then there are individual functional units on that floor. We call them classrooms, right? So from wall to wall, that defines one classroom. So sarcomere, its walls are one Z-disc to another Z-disc. And there are many classrooms in a row on our second floor, just like there are many sarcomeres in a row in a single myofibril. Now let's talk about I bands and A bands, and M line, H zone, and Z disc. Now the I and the A, that just basically stands for isotropic versus anisotropic. Anisotropic means that the band appears dark, and isotropic means that it lets light through, so your eye catches more light. So let's talk about the definitions of these I bands and A bands. First, let's start with I band. This is the light band, and it contains thin filaments only. Remember, the thin filaments are the actin, troponin, and tropomyosin. And the A band is called the dark band. It's in the middle of the sarcomere. It's composed of the entire length of the thick filaments, and on its lateral sides it includes some overlapping thin filaments. So here's how I remember this. The second letter in the word dark is A. And so I remember the dark band is going to be the A band. And you can see that in the histological picture up here on the top right. It looks dark to you. And then I remember the second letter in the word light is I. And that helps me remember that the light streak that you see in this top right picture is the I band. And notice that the I band overlaps from one sarcomere to an adjacent sarcomere, doesn't it? Now what about the M line? The M line is a thin transverse protein meshwork that appears as a dark protein disc in the center of the H zone where thick filaments attach. Now I think of M line for middle. It's just the middle line that we see there. What about the H zone? It's a lighter region in the middle of the A band, in the middle of it, and it contains thick filaments only, no overlapping light bands, or thin filaments I should say, no overlapping. So I remember the second letter of the word thick is H, and that reminds me that the H zone consists of thick filaments only. And then the Z disc, of course, is just the border of a sarcomere, and it contains proteins called connectins, and this is where the I bands can attach to the Z disc. Now, when muscle contraction occurs, that you're going to learn in just a moment, it's important to know which of these bands or zones or lines disappears and which stay the same. So when there is contraction, these are the changes that occur in a sarcomere. Number one, the width of the A band remains constant, but the H zone disappears. The Z discs in one sarcomere will move closer together. The sarcomere narrows or shortens in length, and the I bands narrow or shorten in length. Now let's go through the part of the lecture that I've already taught you in class regarding membrane potentials. Because in order to get muscle contraction, it is very important that the student understands that mechanical events, which is contraction, are preceded with electrical events. Must be preceded. So let's set the stage for this electrical event called an action potential. In this picture, you are seeing the ends of a neuron called the terminal bouton or axonal terminus or synaptic terminus or synaptic knob. It has all sorts of names. And you're seeing these little knobby-like structures. To me, they look like the head of the radius. I hope they do to you too. You can tell they're connecting to some sort of spherical entity in this picture. And they're communicating to the soma or cell body of a downstream neuron. But neurons can also synapse or connect to skeletal muscle. So we need to know how neurons are excitable, and we need to also understand how muscles, in particular skeletal muscle, are excitable. And that word means that they can change their membrane potential. They can destabilize it. And when you destabilize this idea of a membrane potential, what we're really talking about is an electrical current. It's really no different than when you plug something into your outlet in your wall and you flip the switch on. You have electricity going through that wire, an electrical current. An electrical current is flowing through a neuron and skeletal muscle too. We at Saddleback require anatomy instructors to teach this to their anatomy students, because when you get to physiology, we go into more advanced detail. So here we go. Let's establish the idea of, first, a resting membrane potential. In order to appreciate what a resting membrane potential is, we need to first understand intracellular versus extracellular environment. Let me say that again. What do you find inside a cell versus what do you find outside? Please look carefully. In green here, I'm trying to show you a phospholipid bilayer. And on the inside of the cell, dissolved in the cytoplasm, we have electrolytes. That means things that have a charge. Either a plus charge, +2, or a negative charge, -2, -3, but they have a charge. And notice that we find similar things outside of the cell, but in different proportions, different quantities. So it's very important for you to understand, number one, we have this sidedness of the membrane, different amounts of things found on either side of the cell membrane. Another way of saying that is there is clearly a gradient, a high side and a low side for these electrolytes. So there is a gradient. The next thing that you need to understand is the idea of diffusion. There is a law that is related to diffusion and the rate of it, and several things are necessary for diffusion to occur. And diffusion is a net movement of a substance from one area to another area. A net movement. If a substance does not have a gradient, if it is at equilibrium across the cell membrane, does that mean it doesn't move? Of course not. The substance still moves. It will move from one side of the membrane to the other side of the membrane as long as it has permeability, but there will be no net gain or loss of that substance from either side of the cell membrane. If one substance moves from the inside out, well then another partner of it on the outside will move in. There will be equal exchange, but they are still moving. In diffusion, you are saying that there is this gradient, a high side and a low side. And it's not enough just to have a gradient. You need other things as well. And one of these other things that you need, that will be our primary focus, is permeability. If the substance cannot get through the membrane, it doesn't matter if there's a gradient or not, it can't move. That's really important, and most students miss that. You must have permeability. If and when permeability occurs, then the substance will move from an area of high concentration to an area of low concentration in a net fashion. Please look very carefully at the two electrolytes that I have boxed in gray in this picture. Let's look at sodium first. If suddenly sodium had permeability, can you tell me which way it would move? It would move down its gradient. So your word choices should be, it would move from the outside of the cell to the inside of the cell. What about potassium? If and when suddenly potassium had permeability, which way would it move? It would move from the inside of the cell to the outside of the cell. Next concept. Clearly, if we're just looking at sodium and potassium, highlighted in this gray box, they have a chemical gradient. That means they have unequal quantities of that substance across the cell membrane. But you know what else? Sodium and potassium also have a charge associated with them. So now let's redo that question again. If and when sodium had greater permeability, which way would it move? And you would say from the outside to the inside. If I then followed up that question with another question that said, and what charge would accumulate inside the cell then? You should say that the inside of the cell would accumulate positive charges. What if potassium suddenly had, potassium is shown here as the letter K. What if potassium suddenly had greater permeability? I hope you would say it would move, diffuse down its concentration gradient, its chemical gradient, from the inside of the cell to the outside. Okay, follow-up question. What would happen to the charges inside the cell? Clearly, the cell would lose positive charges. And just like in life, if you lose your positivity, what do you become? Very negative, don't you? Same with a cell. If it loses its positive charges, it becomes progressively more negative. Let's do this again and again. Please hear me. In this picture with this blue circle, I'm trying to show you that a cell has sidedness. It has separation of solutes. But it's not just separation of chemicals, it's separation of charges too. And out of all those things that I listed on the previous slide, I want you to know that overall the inside of the cell is actually much more negative than the outside of the cell. There's sidedness of charges. The reason for the inside of the cell being negative is multifactorial, many reasons. But we can reduce it to 3 big ones. And I don't think that's enough, is too much, I should say, for a student to know. 3 main reasons. 1, because of your body pH, your proteins that are abundantly found inside of a cell are negatively charged. That's important. 2, there is a pump on the cell membrane called the sodium-potassium ATPase. This ATPase engages in primary active transport. That means it uses ATP to build gradients. It is responsible for building the high sodium gradient outside of the cell and the high potassium gradient inside the cell. It moves 3 sodium out of the cell for every 2 potassium that are pumped into the cell. Ask yourself, 3 to 2. Is this equal exchange of a chemical? No. 3 sodium out, 2 potassium in. Ask yourself again, is this an equal exchange of charges? No. 3 positive charges leave for every 2 positive charges coming in. That's a net loss of a positive charge. Therefore, the inside of the cell is becoming more negative thanks to this ATPase. We say it is electrogenic. Lastly, the membrane of your cells are on average 100 times more permeable. There are a few leaky channels that only allow potassium to move through them. So here you have the sodium-potassium ATPase, a lot of it, pumping potassium inside the cell, only to have a few leak channels that allow potassium to backwards flow, diffuse out of the cell. That's a loss of positive charges. And that means the inside of the cell is negative. Because of these three things, with the number one contributor being these leaky potassium channels, the inside of the cell is negative. And therefore, every single cell of your body is really set up like a battery. And if you think of your standard battery, you think of one pole side being negative and another pole being positive. So in this picture, I am again showing you the separation of charge. And inside a battery, this picture that I show you in the top left, you have chambers inside of a battery. And they have separation of chemicals, which also have separation of charge. And when you use your battery, the chemicals are allowed to diffuse down their concentration gradient. Similarly, the charges flow down their electrical gradient. And when everything reaches equilibrium, what do you say about that battery? I'll tell you what you say. Oh, my battery is dead. So every single cell in your body has a separation of chemicals, separation of charges, and in a few set of cells, they have the ability for these charges to move and the chemicals to move. They have permeability, but they never will reach equilibrium, because if they do you know what I will say to you? Oh, you're dead. The moment these chemicals and charges reach equilibrium, your excitable cells, like neurons and skeletal muscle, would in fact be dead. You would be dead. So let's talk about some terms. A lot of times students are bullied into submission because they let words scare them. A current. When you talk about an electrical current, that really means just the movement of charge. And opposite charges are attracted to each other. Voltage. You think of a 9-volt battery or some other voltage of battery. What does that mean? It simply means how much chemicals and charges in that battery are separated. The more you have of the separation of opposite charges, the greater the voltage. And another way of saying that is voltage difference. Potential difference or potential. That all means the same thing. When you have a 9-volt battery, you are saying that from one chamber to the next inside that battery, you've got a separation of charge worth of 9 volts. Resistance. Resistance means opposition. If we're talking about current flowing, charges flowing, then resistance would be something that won't let the charges move, right? And in our story, it's going to be cell membrane permeability. If the membrane is not permeable, then the charges can't flow. The chemicals can't flow. That's resistance. Conductance. Conductance means now that permeability has changed, and now the chemical gradient and electrical gradients, these things can flow down their gradients. I like to think of a street light analogy. Okay? The street light is red. Clearly that's resistance. It's opposing your movement through the intersection. You should not conduct yourself through the intersection. But when the light turns green, that means you now have permeability through the intersection. You can now conduct your car through the intersection, and now you're going to be on the other side of the intersection once you drive through it, correct? It's a similar thing across the cell membrane. Instead of street lights, they have proteins with channels, and the channels can be closed or they can be open. Red light, green light. Red light, resistance, no conductance, can't move. Green light, open, can move, and the substance will move from one side of the cell membrane to the other. And when the substance moves that has charge, you now have an electrical current. I can't say this enough or stress it enough. Because of the separation of chemicals and their electrical charges, every cell has a membrane potential. Remember, the word potential means same as voltage. It's like a battery. And we use the word potential because just like a battery that you might hold in your hand, look at that battery and you say, this has the potential to do work for me, right? You could put it in your phone. You could put it in, we put batteries in everything and we flip a switch and then that electrical machine does something for us. Our phone most certainly does work for us when we make a phone call. And when your battery goes dead, it means because of the chemicals and their charges have diffused, reached equilibrium, and now there is no net gain or loss of movement of charges. Your battery is dead. So stop and think. Let's stop and think for a moment. If the inside of the cell is negative and the outside of the cell is more positive, then we can think of charges moving across the cell membrane attracted to each other. The negative charges being attracted to the positive and vice versa. And when, my dears, we have this current flowing, then it has the ability to do work. When something suddenly starts to move, like current moving, this is no longer called potential energy. This is now the energy of motion, and that's called kinetic energy. And we can harvest that and turn it into other kinds of energy in order to do work. Students oftentimes ask me, well, Kara, how negative is the inside of the cell anyway? They want a number. Okay, I'll tell you. It depends on the cell, actually. Different cells have different separation of charges to a greater or lesser extent. You can think of them as a more powerful battery or a wussy battery. So on average, it's about minus 70 millivolts, which is not a very big number, but it's enough. It's enough for many of your excitable cells to do amazing things for your body. All right, so now we've answered the questions down at the bottom of the screen. Why is the inside of the cell negative? Big three. You should know those by now. Which of those is winning? Which of those is the most responsible for the negativity inside the cell? It's the back leak of potassium outside of the cell. Just a slow leak, but it still is the loss of a positive charge. And again, this is still simple diffusion, this backwards leaking. It's simple diffusion. The potassium is leaking through these channels from the inside of the cell to the outside. Now, what if suddenly the cell membrane became more permeable to some substance? Look, in this picture I'm showing you a circle. It represents a circle. And I want to remind you that the outside of the cell has more sodium than the inside. Let's start with sodium. Let's say suddenly permeability to sodium increased. Ask yourself, which way would sodium want to diffuse? It would diffuse from the outside of the cell to the inside of the cell down its chemical gradient. But it would also be pulled into the cell down its electrical gradient as well. Sodium is positively charged, and inside the cell there are negative charges for the big three reasons I just taught you. So sodium most certainly wants inward movement into the cell, down both its chemical gradient as well as its electrical gradient. Now what would happen to the charge inside the cell, the membrane potential, if sodium moved into the cell? The answer is the inside of the cell would become less negative. It wouldn't stay at minus 70 millivolts, which is what I'm showing you on the screen. It would become less negative, progressively more positive. It dropped from minus 70 to minus 50 to minus 30. And students say, well, how much less negative and more positive will the cell become? Well, that depends on how much permeability sodium has and how long those channels stay open, doesn't it? It's situational. It's dependent on how much permeability. So let's talk about these cells that have the ability to tap into the potential energy. Which cells of your body allow the movement of chemicals and their charges to create an electrical current? These are your excitable cells. They include smooth muscle, cardiac muscle, skeletal muscle, even glands, as well as neurons. They have the ability to suddenly and instantaneously, and for a certain amount of time, they can change the permeability across their cell membrane. Why is that? They have different kinds of gene expression. They express genes that lead to proteins that are integral proteins. That means they go through the entire cell membrane. And these proteins create channels. And the channels, like my streetlight analogy, they're gated. That's what we mean, gated. They can open and close, just like a gate opens and closes. So we need channels with gates, and these excitable cells express these kinds of gated channels. Now I want to ask you a question before I move on. Your house is gated. You leave your house and you gate it up. You close it up. When you come back to your house, you don't have permeability through the doors or the walls. You can't just walk through them. You must ungate them. How do you ungate your house? How do you open your house so you can get from the outside in? Do you insert a key into a lock? In biology, we don't say the words lock and key. We use receptor and ligand. Your key is your ligand. It fits into your lock. Your lock is the receptor. It receives the key. So your front door that you use a lock and key mechanism, that's your ligand gated channel, isn't it? How many of you out there get into your house a different way? How many of you use a transponder inside your car that has a battery in it, and then the transponder, you push this button, and there's a receiver inside your garage, and your garage door opens? Well, that's not a lock and key mechanism, is it? No, that's dependent on a certain amount of signal being received by your receiver inside your garage. And I'm here to tell you that if your transponder battery runs out of voltage, goes dead, can you open your garage door? Nope, you can't. So that's a voltage gated channel, isn't it? You need a battery, and you need to send enough electrical signal to your receiver to get it to open. Your cells that are excitable, they have these two gates as well. They've got a ligand, lock and key, and they've got a voltage gated channel where a certain amount of electrical current must be flowing to get the garage door to open completely. Let's continue on. So, ligand gated channels, lock and key. For anatomy students, they should just know two varieties. Two. These two varieties are sodium ligand gated channels. And these sodium ligand gated channels can also be given different names. They can be, in these sodium ligand gated channels, just to back up, that is a name to describe which ion is allowed to flow once it's opened. Now, the other classification, that's what I mean by I want you to know two. I want you to know the chemical classification versus the other classification that tells you what substance, what key unlocks the gate. And that's acetylcholine. So, acetylcholine receptors, when they are unlocked, they're going to allow sodium to flow. Now, acetylcholine receptors, that word means that the lock is unlocked by the key called acetylcholine. When the acetylcholine binds, then sodium will flow through that channel. There are a couple of subdivisions, subcategories of acetylcholine receptors. There's nicotinic and there's muscarinic. Now it turns out nicotinic was called that because it was a different chemical that unlocked this channel. Think of it like a skeleton key. So they applied nicotine onto this receptor and it unlocked the channel as if nicotine were acetylcholine itself. That's part of the reason why when people smoke they get the fine muscle contractions. It's because the nicotine in their bloodstream is unlocking these channels and current is flowing, and then they get muscle contraction as a result. With muscarinic acetylcholine receptors, scientists discovered that this toxin from a kind of mushroom also unlocked this other variety of acetylcholine receptors. Now, you may or may not, depending on your anatomy teacher, learn more about muscarinic acetylcholine receptors, but for my class, you certainly will be abused with that more in lecture. Voltage-gated channels, like I said, they unlock when there's a little bit of electrical current flowing through the cell membrane, like you pushing on your transponder to get your garage door to open. How many of you are like me, where you try to see how far away you can be from your house and push the button and get the garage door open so that you don't even have to stop and wait for the garage door? You could just go Dukes of Hazzard style up your driveway and into your garage. If you're too far away, you don't get enough signal to your receiver, do you? So voltage-gated channels, they have to get enough signal, electrical current in this case, for them to open. And they are named, very similar to the ligand-gated channels, for the substance that they allow to flow through them. So you have sodium voltage-gated channels. That means when the voltage-gated channel opens, sodium's allowed to flow. You have potassium voltage-gated channels. That means when that voltage-gated channel opens, potassium gets to flow. And you also have calcium voltage-gated channels, which I'm sure you know by now, means when that channel opens, calcium will flow through it. In order for us to appreciate the location on a neuron and skeletal muscle cell of ligand-gated channels, lock and key, versus voltage-gated channels, we should explore the anatomy of a neuron. Looking carefully at this picture to the right, you see the cell body, which is also called the soma. It surrounds the nucleus. The cell body surrounding the nucleus is oftentimes referred to as the perikaryon. Peri meaning around, karyon is an old word for nucleus, so around the nucleus. Notice that it has branches that are extending off the cell body, short branches. And these are dendrites. Hey, dendrites, that's where you're going to find your lock and key channels, your ligand-gated channels. Then notice you have one long extension off the cell body, and this is called an axon. And then at the end of the axon, you have the nerve terminal. It's also called the axonal terminus, or synaptic terminus, or terminal knob, or terminal bouton. I will refer to it as a terminal bouton. In the picture, it calls it a nerve terminal. All along the axon and terminal bouton, we find voltage-gated channels. On the axon, you find sodium, and as well, you find potassium voltage-gated channels. On the nerve terminal, or terminal bouton, you would find all three: sodium voltage-gated channels, potassium voltage-gated channels, and calcium voltage-gated channels. I encourage students who are listening to this to take this picture and label it for the different kinds of channels you would find in each location. Again, dendrites and soma, ligand-gated channels. Axon, sodium voltage-gated channels and potassium voltage-gated channels. Nerve terminal or terminal bouton, sodium, as well as potassium, as well as calcium voltage-gated channels. Dendrites, as I said in the previous slide, are the receiving side of a neuron. They are going to have ligand-gated channels. The soma, it is going to do a lot of jobs for a neuron. It contains the nucleus, so therefore there will be a lot of gene expression. And the soma is going to be responsible in most neurons to manufacture neurotransmitters. These are chemical signals. Neurotransmitters are the key that binds into the receptor. Neurotransmitters are the ligand that binds to ligand-gated channels. The acetylcholine that I referred to in the previous two slides is a neurotransmitter. It is the chemical ligand or key to unlock the gate. The axon is an extension of the neuron. It's going to help conduct an electrical signal. It's an electrical current. This is called an action potential. Don't let the word bully you. Action potential simply means an electrical current. Many neurons of your body, their axon is wrapped around a very fatty, or I should say it's not wrapped around, it is wrapped by a very fatty material from another cell. And you'll learn more about this in your nervous system unit. But this other cell with its phospholipid bilayer wraps around and around and around and around the axon, and this fatty bilayer ends up creating something called a myelin sheath. You will learn more about that later, but this myelination allows the electrical current, the action potential, to travel faster, a faster rate of transmission. Axon terminals, synaptic terminals, terminal bouton, nerve terminals, those are the end of the axon. And this is where we find all three of our different kinds of voltage-gated channels, but this is also where the neurotransmitters of this neuron will be released. And they will be released into this space, this white space below the nerve terminal, and they can diffuse and reach downstream neurons and unlock their ligand-gated channels. There's an important piece of the neuron. It's the transition between the soma and the axon. This piece that transitions between soma and axon, this is called the axon hillock. This is also where, quote, an all or none decision is made. All or none decision. Oftentimes it's referred to as the trigger zone. For example, if I were to shoot one of my students, and I love my students, so therefore I'm going to shoot this student that I'm thinking of because I like her so much, I'll pull the trigger slowly. Slowly. Does that make sense? It shouldn't. Because even if you've never fired a gun, you should appreciate that there is a trigger mechanism on a gun. And that means it doesn't matter how fast or slowly I pull on the trigger. Eventually I will get to the trigger point and that bullet will fire. And it will fire with the same force regardless if I pulled quickly or slowly. The neuron is the same way. It's just like a gun. You want it to fire an action potential, you want it to send an electrical current, then it's an all or none. You either pull the trigger or you don't. It's all or none. And this all or none decision, the trigger point, is at the axon hillock. Students say why? And I respond with, this is where you no longer have the ligand-gated channels, you have voltage-gated channels. Now, side note, those of you who have more advanced training, you just heard me say, at the axon hillock we no longer have ligand-gated channels, we have only voltage-gated channels. And I am speaking in absolutes for testing purposes. I know that there is a mixture of these found in different places of the neuron, but it is true to say that beyond the axon hillock, through the axon to the terminal bouton, voltage-gated channels are the most abundant, and that's why I am speaking in absolutes. So what's the trigger point, this all or none decision? We don't call it a trigger point, we call it threshold. Same kind of concept. And what it means is, in order to get those voltage-gated channels, remember, they need a certain amount of electrical current to get them to open. Well, the inside of the cell has to go from roughly minus 70, become less negative, more positive, to about minus 55. And again, different excitable cells have different ranges, but this is a typical one. So you need about a 15 millivolt change, becoming less negative, more positive, for them to open. You don't get that minus 15 millivolt change, then they don't open. All or none, threshold. In order for a nerve signal, an action potential, to be transmitted between two cells, it must pass down an axon, but then it has to cross across this gap between the two neurons. You see, neurons can orient from soma to axon to terminal bouton, and then you have the next soma of the next cell. I like to think of them as dead stick figures that are lined head to toe. So clearly the electrical current has to go from one to the next, but how do you get an electrical current to travel through space? How do you get an electrical current to jump from your wire through the air of your room and into your phone? That's not how it happens. So when you're talking about sending signals between two neurons, or a neuron and a skeletal muscle cell, the electrical signal has to be transferred into a chemical signal, which in turn, the chemical signal can bind to your ligand-gated channels on the next cell, which can then turn back into an electrical signal. We call this an electrical-chemical transmission. So we're talking about an action potential, that's the electrical part, and then the synaptic transmission, that's the chemical part, the neurotransmitter that's released. So down now on this screen, you're seeing a terminal bouton shown in red from several other neurons synapsing with a dendrite. Remember, that's the receiving end of a neuron. So at the terminal boutons that are shown here in red, that's where you're going to have neurotransmitters. Now, if you're talking about the terminal boutons shown here in red, you can see the space in between the red terminal boutons and the gray dendrite. That space is called the synapse. So the red terminal boutons are pre-synaptic. They come before the synapse. And the dendrite, well, that's part of the post-synaptic cell. So the terminal bouton is going to release neurotransmitter, which will diffuse across the synapse and bind to the ligand-gated channels on the next downstream postsynaptic dendrite. This is what we call transmission, synaptic transmission, or sending the message across the synaptic cleft. That's the space between one cell and the next. When we come back for our third segment, we will now talk about a special kind of synapse between a neuron and skeletal muscle cell.